Hi guys, so let's start with analyzing a pedigree, right? So let's understand autosomal disorders, all human gene disorders, and see how analysis of pedigree is done. So when we start analyzing a pedigree, there are three things that we should do. The first thing that we should do is understand if there is a sex bias in the pedigree. Okay? So what is a sex bias? So it is to check if the disease happens more to one sex. So either males have more incidences of the disease or females have more incidences of the disease, right? So let's look at this family. Let's number the pedigree. This is generation 1, generation 2 and generation 3. So individual 1, 2, 1, 2, 3, 4, 7, 8. Right? So you have a total of 8 plus 2, 10 plus 11, 21 individuals in this family that are on the pedigree. Correct? In 3 generations. Out of them, let's see how many males, out of 21, how many males have the disease? So 1, 2, 3 and 4. 4 out of 21 males have the disease. And how many females out of 21 have the disease? So let's say 1, 2 and 3. 3 females out of 21 have the disease. So see, in 21 people, 3 to 4 people, 3 to 4 females and males have the disease. So because the numbers are close, we can say there is no sex bias. If the number had varied by, let's say, 10 to 1, then there would have been a clear sex bias, right? So we will notice from here on that autosomal disorders usually don't have a sex bias. Sex biases are present in sex-linked disorders. Okay. Moving on, the next thing that we check for is known as a carrier mark. So who is a carrier? A carrier is an organism or an individual who carries faulty gene but is unaffected. So in a family or in a pedigree, they will not be hashed. They will look like normals, right? Look at these 2 individuals. They are both normals, but when they have a kid, the kid will have the disease. So this is a classic carrier. So what is a carrier mark? What is the mark of a carrier? The mark of a carrier is 2 healthy parents having a diseased kid. You will see, and I'll explain why, recessive disorders can carry, usually have carriers, whereas dominant diseases or dominant characters, they never have carriers. So let's understand why, and to understand that you will have to come to the third point of a pedigree analysis, and the third segment of a pedigree analysis is understanding genotypes. Let's start with autosomal dominant kind of diseases. Okay, autosomal dominant diseases, for example, this is for Huntington's. Now in Huntington's, the affected chromosome is chromosome number 4. In chromosome number 4, let's say there are 2 alleles. The capital D dashed allele is the affected allele or the abnormal allele, whereas the small d is the non-affected or the unaffected or the normal allele, right? So capital D dash is the diseased allele, small d is the normal allele. Then genotypic combinations could be a homozygous dominant or a heterozygous dominant. But if even one copy of the affected allele is present, as it is dominant by the law of dominance, it will be affecting the organism. Am I right? So for both these genotypes, whichever genotype the organism the human has, they will be affected or diseased. However, only in a homozygous recessive state, because it's small d, right? So both the alleles are unaffected alleles. Then only you can have a normal unaffected individual, right? Now let's come down to the pedigree and understand what I'm trying to say. This individual is diseased, right? So it will always have a capital D allele with it. Now the next allele can be capital D or it can be small d, right? We don't know which one. This one is unaffected. So it can never have a capital D allele because capital D is the dominant allele. Therefore it will be small d, small d. Now let's make a punit for generation 1. So you have capital D dashed, small d, small d. This is one combination. In this combination, you would see that all progenies will always get a capital D dash from the father, right? So in this case, all progeny will be affected. The other cases where capital D dashed and small d is with small d crossed with small d, small d. In this case, however, you will see that 50% will be affected. That is the prediction, right? And 50% would be normal. Now, let's look at what happens to this couple. To this couple, they have had a diseased daughter, a normal son. A diseased daughter, a normal son, and a diseased son. Right? So, because they have had both normal and affected children, therefore, we can very easily say that this is not their genotype, the father's genotype, because then these kids were not possible. Small d kids were not possible. Am I right? So from this, we can deduce that the father is capital D, small d. Now, then this kid is small d, right? Let me write it inside. This is small d. This kid is also small d. The ones that are diseased are capital D, small d. They cannot be capital D, right? This is the only possible combination as the punit has told us. So these are all capital D, small d. Again, the normal individuals are all small d. And similarly in the next generation, because the cross stays the same again. It's almost identical. We do not need to check for sexual X or Y linked preferences. Therefore, the same cross will go on for Gen 2 as well, where you would get these are all small d individuals. And this is the capital D, small d individual. Right? So that is how you solve an autosomal dominant pedigree. Now we will move on to the recessive autosomal pedigree. Moving on to the recessive autosomal pedigree. I told you recessives can have carriers. So same thing we will consider. We will consider 2 alleles for a chromosome. Capital D allele in this case is the allele which is the unaffected or the normal allele, whereas small d is the allele for the affected, right? However, because this disease is recessive, in a homozygous dominant case, the organism will be unaffected because there is no allele for the disease present. In this case, capital D, small d dashed. These organisms have one normal allele, one allele for disease. The normal allele which will show the phenotype, right, will have the expression, phenotypic expression as it is dominant. However, in this condition, which is the heterozygous dominant condition, because it carries the allele for the disease, these organisms will then become carriers. Because they carry the allele, but their phenotypic expression is of that of the dominant, that is unaffected. So then they become carriers. Do you understand why then dominants can never have carriers? Because in this case, in the heterozygous dominant state, because the dominant allele, which is the diseased allele, is already present, the organism will have the disease, right? But here, because D is normal, capital D is normal, small d is the problem, and it is recessive, the capital D will show the effect, right? However, they are able to pass on the small d to the next generation, and therefore they are carriers, they carry it, okay? And only small d dash would give you the affected organisms, correct? So now let's do the genotypic placements. This organism has to be small d dashed, small d dashed. This organism has to be also small d dashed. This is the for sure. This person can be capital D small d or capital D, correct? Or capital D small d. Now let's look at what the punit says, because for both of them it will be similar. We'll do 1. So capital D small d crossed with small d small d. Let's dash the d's because it's a diseased condition. And the second cross that we have to do is capital D crossed with small d dashed, small d dashed. Now in this case, all combinations will come out to be heterozygous. And if it is heterozygous, then all progeny would be what? All progeny would be carriers. So the disease should not show in the family for that generation. However, here you can have a 50% chance of the disease. So 50% affected, 50% would be normal. Correct? So now let's see if these couples have had any diseased child or not. For this couple, generation 1, couple 1 and couple 2. For generation 1, couple 1, they have had in the second generation 1 son and 2 daughters, all of them healthy. So for them this has worked, correct? So could be any of these, but most probably this has worked where all of them are carriers. Yeah, so it's impossible to say which one of these it is, because again I told you this is just a prediction. It could just be that this woman, she is a capital D small d dash, she is a carrier. It can be possible, but they were just lucky to get all normal kids. Or it could be so that she was absolutely okay and therefore they had all carrier kids. There is no way of knowing unless we do a genetic profiling, right? However, for this couple, the same thing is true. It could just be that the male was a carrier and they were unlucky to have the kids, one kid who is diseased, or there is no other option, right? So in this case, we can say that the male was 100% a carrier because they have had a diseased son. But for them, it's impossible to say. Okay? Now, moving on to the next generation, let's see. This is a small d dashed case, and this is a capital D small d dashed case, let's say, because capital D though is not possible, right? So it's obviously a carrier. Yeah. So here again, this is what has happened. Now in this case, this person is a carrier, this person is a carrier. So let's make crosses for 2 carriers. So capital D small d dashed, capital D small d dashed. So you will have normal individuals, possible. You can have carrier individuals, possible. Or you can have diseased individuals, that is also possible. So for them, that is what has happened, right? So they have either carriers or normals, but they obviously have a diseased. And if both parents are diseased, then obviously the disease will be present in all the kids. One thing that has to be seen in this particular pedigree is that a disease can skip a generation. So in generation 1 for couple 1, the father has the disease. But in generation 2, none of the kids have the disease, right? And the disease resurfaces back in the third generation. So another interesting thing about recessive disorders is that it can skip generations, because mid-generation there can be a carrier condition. I hope this much was clear. We will move on to sex-linked disorders post this. Please go through this because sex-linked disorders can get more confusing to understand. We will continue with this in the next video. Thank you.